doctoral at the Biological Clocks group at the Max Planck for Evolutionary Biology. And today I'm going to talk to you about magic traits. So, so in 1943, when Ernst Mayr defined biological species as groups of interbreeding natural populations that are reproductively isolated from each other, he underscored the central role that reproduction plays in what makes a species a species. Therefore, understanding speciation is largely a matter of understanding the barriers to reproduction that exist between diverging populations. And through a lot of work, it's become apparent that a certain class of barriers called magic traits seem to be the ones that are most likely to result in speciation. So a magic trait is a trait that plays a role in both the ecology and the mating of an organism. And more specifically, it's an allele or set of alleles segregating under ecological divergent selection that results in one or more barriers to reproduction. Classically, mate signaling traits have been examined as potential magic traits, with one of the most famous examples in heliconius butterflies, where the color of the wing patterns serves a role in Mullerian mimicry, while also allowing individuals to recognize potential mates. And similar examples have been seen in poison dart frogs or the African weakly electric fish, which use electrical signals to find prey as well as mates, or these coral reef hermits, which use their color patterns to find mates, but also camouflage. However, a separate class of traits called the automatic magic traits have received far less attention, and these are traits that assort mating in time or space and thus result in reproductive isolation sort of automatically. And examples of these include habitat preference, like for the Ragoletus fly, which can breed on either apples or hawthorn berries, or timed reproduction, where populations reproduce at different times of the year and thus miss each other. And these automatic magic traits have been suggested to be the most likely traits most likely to result in speciation. And moreover, especially for timed reproduction, it's very common for organisms to time their reproduction across the tree of life. Here's just a little phylogeny with all of the known lunar spawning rhythms, but you can also think about flowering times in plants or different insects breeding at different times of the year. So given the likelihood of these traits in producing speciation, as well as their apparent commonality, these automatic magic traits might actually be very important diversity generating mechanisms. But in order to assess their importance, it's good to first ask, what is the efficiency of the magic trait? Because if a magic trait is not very efficient at reducing gene flow, then it's not going to be very important for driving speciation. And a lot of studies have shown that it's very rare for a single trait to be enough to complete the speciation process. Rather, it's the coupling of multiple traits that evolve over the speciation process that sort of clench total reproductive isolation. And another access to this is that different genomic architectures might be more amenable to coupling. For example, if the trait under divergent selection is more polygenic, then the greater number of genes in the genome, the more likely it is that one of these genes is going to be next to a reproductive barrier and thus sort of catch the reproductive barrier, allowing for this coupling to occur. So to summarize, magic traits link ecology and reproduction, which allows speciation to happen, and the automatic magic traits are suggested to be the most amenable or the most likely to produce speciation. And for an automatic magic trait to be important in speciation, it must be efficient and amenable to coupling. And so an automatic magic trait with sort of like a favorable architecture is probably one that's highly important for generating diversity. So I wanted to share with you guys an example of an automatic magic trait in the species that I study, which is Clunio marinus, marine midge. And we're interested in Clunio because it times its life cycle and reproduction to the lunar cycle using an endogenous biological clock. So Clunio spends the majority of its life as a little larva, a little worm living in these algal tufts at the bottom of the intertidal zone. And these tufts are only exposed during the lowest of the low tides, which happens around new or full moon. And during these tides, the individuals will emerge to the surface of the water in a close, and then the adults will sort of swarm on the surface, and the males will find the females. And then the male will take the female to a close by patch of algae, like you can see in this video, where the female will then lay the eggs in the algae and the larva will hatch. And the female really needs the algae to be exposed so she can glue the eggs down before the incoming tide washes them away. So there's some quite strong selective pressure on the timing of emergence so that she has access to these substrates. So you can imagine if there were somehow to be some divergent selection, this could quite easily turn into an automatic magic trait. And indeed, in two places in the Brittany Peninsula, we find these multiple sympatric chronotypes, which are sort of subpopulations which have diverged in their reproductive timing. And a genetic study found that these chronotypes are actually the result of a single recent colonization event, and that there's very little genetic differentiation between them, which suggests two things. Either one, the timing barrier is actually a very inefficient barrier and allows for a lot of gene flow, or that divergence occurred very recently. So this sort of leaves me with three questions. The first is, how important and efficient is Clunio's reproductive timing for reducing gene flow? Next, what other barriers to reproduction might exist if the timing barrier isn't very efficient, if it's a little bit leaky? And lastly, what loci might underlie these different barriers? So to answer the first question, we went out to the field in Roscoff where there are two chronotypes, a full moon emerger and a new moon emerger. And over the course of 2 lunar months, we sampled individuals that were mating with each other. So they were a copula, like in the picture here. And we took those individuals back to the lab and designed a multi-locus genotyping assay to genotype highly diverged regions, which we located with FST. And basically, by doing this, we can sort of assign each individual into one or the other chronotype, or if it's some sort of hybrid, we'd be able to tell. And our expectation for if it's an effective barrier is that the genotypes will pretty much correspond with the time of the month, and that the genotypes of the couples will match each other. So when we look at the data here, with the x-axis being the day that we caught them and the y-axis being the time that we caught them, we see, first of all, that the two chronotypes are actually separated in time by when they reproduce. So the full moon individuals are emerging during the full moon, the ones in yellow, and the new moon individuals are emerging during the new moon, and there's a nice gap in between the two. But what we might also notice is that there are apparently a lot of hybrid individuals here with sort of intermediate colors. So to better quantify the level of gene flow, we sort of classified individuals based on their genotype. If it was greater than 0.5, we said that it was a full moon, and less than 0.5, we said it was a new moon. And from that, we could calculate how many individuals were emerging contrary to their genotype, and we calculated that it's about 6% of the population emerges opposite to what we would expect based on their genotype. And then to sort of quantify gene flow a little bit better, we looked on the couple level, and we found that about 8% of the couples that we caught actually had genotypes that were opposite to each other. So these were like a full moon male mating with a new moon female, or vice versa. So to answer question one, it appears that this reproductive trait, or the reproductive timing, is important, though it's about 8% leaky. So what other barriers to reproduction might exist to sort of supplant this leaky barrier? To answer this question, we did a series of laboratory crosses where we crossed full moon to new moon individuals in the lab to sort of check for reproductive barriers along the life history that you can see on the screen here. And the first place we looked is at the stage where the male meets the female to see if there's something other than reproductive timing that would prevent opposite chronotypes from mating with each other. And to do this, we put a male and a female in a little arena like this, and we timed how long it took for them to mate. And the expectation here is that if there was some sort of assortative mating outside of the timing, then the between chronotype crosses, so like a full moon to new moon or new moon to full moon, will take longer because the male will be somewhat reluctant to mate with the female. And when we look at our data, that's exactly what we see. So here we use Cox proportional hazards regression, and we find that crossing type is actually a significant predictor of how long it'll take for a cross to happen. And interestingly, the interaction between crossing type and female age is highly significant. And you can see this effect in the plot over here for the within chronotype crosses. There's not a huge difference in how long it takes to copulate between if the female was old versus young. And by old female, I mean that the female had emerged and sort of sat around on the bench for a while before we added her to the crossing arena. However, for the between chronotype crosses, you can see that there's actually a huge difference when the female is old, where the males then become very reluctant to cross with the sort of opposite chronotype female. So this is evidence that there is actually assortative mating happening here outside of just the lunar timing and the circadian timing. So next, we're curious about the sort of fertilization stage. So we're wondering if there was some sort of gametic incompatibility, then we'd sort of see a reduction in the rate of fertilization. So we counted the number of fertilized eggs that we got from these different crosses, and we found that there actually wasn't a difference for the between versus within chronotype crosses. So we can rule out there being a barrier here. And then next, we're interested to see if there's some sort of hybrid depression. And this would be like the larvae of hybrid individuals wouldn't survive very long or wouldn't be able to hatch. And so our expectation was that if there was hybrid depression, then there would be a decrease in hatching rate for the between chronotype crosses. But again, that's not what we saw. We actually saw the opposite. So we can rule out there being a barrier here as well. And I'll get back to that last question mark in a second. So to come back to our questions, what other barriers to reproduction might exist? Well, we found assortative mating. But what loci might underlie these different barriers? So to answer this question, we went back to the field couples and we started to think about what we had actually genotyped for our assay. And what we did is we picked these regions of high divergence, which are theoretically regions of the genome that are more resistant to gene flow. And they would be more resistant to gene flow if there was some sort of gene there that was responsible for a barrier to reproduction. And so using that perspective, we could basically ask, okay, which of these different loci that we genotyped are associated with the different barriers to reproduction that we found? And so we answered this question using multiple regression. And I'm going to skip over a lot of statistics here and just tell you the results. So basically, we found that several loci were involved for the three barriers that we knew about: the circulunar timing, so what day they came out; the circadian timing, which is what time they came out; and the assortative mating that we saw in the lab. And then, like in the lab where we didn't see any gametic incompatibility or hybrid depression, we also didn't see that for the field couples. But the last barrier, hybrid sterility, we couldn't test for in the field because we couldn't test for in the lab because we didn't have any hybrid individuals. But in the wild, if you remember from that plot, we actually found a lot of hybrid individuals. So we could basically test to see if hybrid females had a reduced number of eggs. And we found that two loci were actually significantly associated with the number of eggs that a female produced. So if she was a heterozygote at those two loci, she produced fewer eggs. So there's actually a fourth barrier to reproduction here. And if we assess all of these barriers together, there's sort of two observations that we can make. The first observation is that for each locus that was associated with one of the barriers, many of them were actually associated with more than one barrier. And so it appears that there's some sort of like extensive pleiotropy, or maybe the genes are very closely linked together that are controlling these different barriers, which is rather interesting. But the second observation is that every single barrier locus that we sort of found, every locus that was associated with the barrier was associated with at least one clock barrier, which sort of suggests the following scenario: that there was divergent selection happening on Clunio's clock, which resulted in the automatic assortment that we would get from an automatic magic trait. But in addition to that, through pleiotropy, or maybe there are some very tightly linked genes right next door, we actually got assortative mating and hybrid sterility almost as a byproduct of the divergence in Clunio's clock. So to answer our last question, what loci might underlie these barriers, we found that the clock loci and the other barrier loci were actually one and the same. And to wrap this all up, we can conclude that Clunio's reproductive timing was actually very important for driving speciation. So to put this in a more broad context, we knew already that Clunio's clock was omni or polygenic. And we know that these polygenic architectures are more likely to catch barriers and promote coupling. Moreover, clocks in general are known to be polygenic as kind of a feature of these clocks, since there are like several genes which interact with each other in these complicated networks. And we also know that timed reproduction through clocks is very common across the tree of life. And if there's some divergent selection on the timing of reproduction, then you could end up with an automatic magic trait. And so this leaves me with one last question, which is, how often have clocks driven speciation events? And I think that given what we've seen in Clunio marinus, the answer to that question might be more often than we expect. So thank you very much. And if you have any questions, my email is there as well.
Great. Great. Thanks very much, Alexander, for a lovely talk. If you have any questions, please put them in the chat or the Q&A for Alexander, and we'll move on to our next speaker. Harshavardhan, if you want to share your slides whenever you're ready.
So that's weird. Are you able to hear me okay, though?
Yes, absolutely.
Just going to figure out why my computer is doing that.
There you go.
Awesome. Thank you for having me here. I'm an experimental evolution biologist who works with selection experiments, specifically with Drosophila melanogaster, and I'm really excited to take you through some of the highlights of this male-limited selection experiment we've been working on to study intralocus sexual conflict. Like many evolutionary biologists, I'm interested in the maintenance of genetic variants for fitness. The very simplistic picture is that fitness is by definition under directional selection. So in this kind of cartoon situation that I'm depicting here, where you have some distribution of trait value that's mismatched from its fitness function, we would imagine that the fixation of beneficial variants not only shifts the mean of the trait values, it also compresses the available heritable trait variants. In other words, if natural selection is the engine that takes traits from point A to point B in the trait space, heritable trait variance is the fuel that it burns along the way. And an important paradox is that in spite of us expecting the tank to therefore go empty, we always seem to find more and more fuel. There's always a certain amount of heritable trait variance for important life history traits and traits associated with fitness. Now, it's not as though we're without answers for these questions. We're aware that processes like the mutation selection balance, selection acting at a polygenic level, or forms of balancing selection like spatiotemporal selection gradients and frequency-dependent selection play a role in maintaining heritable trait variants. Myself, I'm specifically interested in whether sexual conflict can contribute to the maintenance of some kind of bimodal distribution of traits, as pictured here, through a genotype by sex interaction, where traits that are or alleles that are beneficial in one sex are detrimental in the other, and so on. Now, is there any reason to believe this exists? We're very aware that the traits typically seem to be undergoing a lot of divergent selection pressures. This is evident from the amount of sexual dimorphism that we see typically, and also kind of theoretically from the anisogamy inherent in sexually reproductive organisms. However, in spite of these divergent selection pressures, we're aware that they have a shared genome and a predominant, the majority of traits that are expressed by the two sexes are non-independent. They share positive intersex genetic correlations. And these two factors combining together should give us the ideal conditions we need to see these kind of genotype by sex interactions in fitness. And this is really well exemplified by the Bank-Wohl and Suzanne Mills and team in 2012, when they selected for and against high levels of testosterone in these animals. And they found that males selected for high testosterone had high fitness as against those selected against high testosterone, which had low fitness, and the opposite in females, where selection against high testosterone produced high fitness individuals and selection for high testosterone produced low fitness individuals. So then the question kind of became, perhaps we have these kind of traits that are being modulated by intralocus sexual conflict, as we define this phenomenon. And these are producing these kind of bimodal trait distributions, which maintains heritable trait variants within these populations. Now, ideally, if we want to assess this hypothesis empirically, we want a system where we can take away sexual reproduction without taking away all of the additional facets of recombination and gene shuffling that also contribute to levels of heritable trait variants. We're interested if the sexes alone do something and not so much about the shuffling of genes. So ideally, what you want is to take away one of the two sexes from your population and then look at what happens downstream of it. And trivially, what we would expect to happen is that the sex that's still being selected, the males in this specific cartoon, have all of the trait values move towards the trait space that's beneficial for the males. And this should happen in both males and females because the intersex genetic correlations between the traits in the two sexes are being unchanged. Now, using a bunch of genetic markers and cytogenetic tools, we're able to pull this off in Drosophila because it lacks recombination in the males, but it still can happen in the females and so on. And with that, we we conduct this selection experiment where we make two critical predictions. If we select on males alone without allowing for selection on reproductive fitness in females, we would imagine that there is increased male fitness that's happening at the cost of reduced female fitness. And in addition, the heritable variation available for male fitness should compress drastically as if under directional selection as expected. So briefly, without giving you much of the details about how we do the selection, we did the selection in triplicate. We ensured that we had kind of replication at the levels that we were interested. And I'm going to kind of keep these colors constant throughout the presentation. So I have controls or matched controls, MCs, as I call them, and they're consistently going to be in orange throughout this presentation. And I have male-limited populations or MLs, which are consistently going to be in blue. What you see in the bottom left of your screen is kind of a karyotype representation. Specifically, I'm representing the X and the Y chromosomes along with a pair of 2 autosomes and a pair of 3 autosomes and so on. You'll notice that the male-limited animals are also a little weird looking. They're heterokaryons rather than homokaryons, which is how we would typically imagine our animals, but that's something that will be addressed down the line. And so we conduct selection as is displayed here. Between days 0 to 12, they're pretty much just developing and eclosing from their pupal shells and so on. At that point, we pull the male-limited animals away from the females that they're developing with, the males specifically. We identify the specific ones, put them in with something known as a clone generator female, and collect offspring from there and so on. Given that these animals look a little different from their controls, it's important for me to talk about the conditions in which I'm testing all of the things that I'm interested in. So we create these heterokaryons that are partially male-limited haplotypes and partially derived from the controls, which we then subject to testing. And we do all of our testing for things like reproductive fitness and the like with competitors, which are marked with recessive eye color markers for phenotyping purposes. So earlier I told you that I was making two critical predictions. The first, that male fitness would increase at the cost of female fitness. And the second, that heritable variation for male fitness should reduce over the period of selection. So to assay fitness, we conduct these competitive fitness designs to as nearly mirror their selection conditions as possible. Essentially, we take 30 target animals and combine them with 30 competitors and put them in with 50 of the opposite sex, hold them together for a 48-hour window, which is the amount of male-female interaction that we actually see under selection conditions, collect eggs from them after, phenotype the offspring, figure out which sire or dam contributed what amount of offspring to the population. And to assess the heritable variation, we do something known as a hemiclonal analysis of fitness. Now, a hemiclone is basically half a clone, literally, and we take these haplotypes and we put them under a number of randomized genetic backgrounds, and we assess the heritable variation or the contribution of genetics to the proportion of variance that we see overall coming out of these families. And so kind of going backwards across our results, the hemiclonal analysis of fitness showed that we had a sharp reduction in heritable variation for fitness in our selected animals. And we also had a sharp increase in fitness of our selected males by 21%. And the amount of reduction in genetic variance was actually rather substantial. It was over 3 times, well, it was under 3 times what we were seeing in the controls. And this was a really cool result. We were very excited by it. We thought that we had demonstrated either for the first time or for one of the very rare times empirically that intralocus sexual conflict was in fact maintaining heritable variation for fitness in these populations. However, our competitive reproductive fitness assays were saying something different. So I'd like to direct your attention to the panels C and D below first, where you would see that female fitness assays demonstrated that our controls were much fitter than our male-limited haplotypes, which was along the lines of what we were predicting. And it's a good sign that the selection experiment is kind of doing what is expected, because we're seeing this sharp reduction in female fitness. However, when we shifted to male fitness, we found that there were no differences that we were able to identify between the matched control animals and the male-limited animals, regardless of the number of times we repeated the assay. And we were rather confused by this. So essentially what we were seeing at this point was that our first prediction, kind of the housekeeping prediction that male fitness would increase in the selected haplotypes along with male-limited selection was not quite coming through. We were still seeing the reduction in female fitness as expected. However, prediction 2, that we would see this reduced genetic variance for male fitness, was coming through. And we had also seen this contradiction between our competitive reproductive fitness assays and our hemiclonal analysis, because the hemiclonal analysis was also telling us that male-limited males anyway, were more fit than their controls. And so this contradiction actually gave us some insight into what was actually happening under the hood. And one of the reasons for this is that our hemiclonal analysis of fitness was a much more complicated design to carry out compared to our competitive reproductive fitness assay. And what we had produced as test animals were not true heterokaryons. You'll notice that I have a haplotype of an X23 chromosome combination in blue, which is the male-limited haplotype, combined with control-derived autosomes. But we were forced to introduce a Y chromosome that was from the selection conditions of the male-limited animals. This is not true of the competitive reproductive fitness assays, where we were able to create much more appropriate test animals. And based on that difference, we started to wonder if there were alternative sources of selection that were underlying our selection experiment. One of these is pretty well known. A number of male-limited selection experiments have been carried out in the past, and they typically acknowledge that we arrest female coevolution. We have this interesting situation where we pull these clone generator females from an alternative population into our selection experiment generation after generation, and those females are unable to co-evolve with the males that they mate with, but the males consistently co-evolve to kind of be extremely ideal partners for those females. So perhaps there is selection responses along that axis. The second thing that we were also interested in, because we saw this difference of a Y chromosome, has been addressed less often in male-limited selection experiments, which is that the genetic background in which the selection actually takes place is rather distinct from typical control animals. And specifically, we call this a home coat condition, where we have a novel cytotype, translocated autosomes, shared Y chromosomes, and so on. So we took it upon ourselves to create a better assay of fitness with a fully factorial design that accounted for the females that we were using, for the genetic backgrounds that we were using, and so on. And what we identified was that our selected haplotypes were performing much better than the controls against both females when they were tested under these home coat genetic background conditions. So we were seeing significant adaptation. It was just local to the very specific conditions in which we were doing the selection. And so you see this extremely positive slope going from control to male-limited animals in home coat conditions, not so much under what we call wild type or control-derived conditions. And so what we found was there were two significant interactions, first between selection and background, where we saw this steep increase in the home coat, not so much in the wild type, but also between female and background, where these male-limited animals seem to be showing some amount of adaptation to the females that they had co-evolved with as well, resulting in this interaction plot here. So to kind of wind this up, I want to give you some of the findings from what I talked about here and some of the expanded universe of this selection experiment as well. We had evidence for adaptation to these alternative sources of selection, which we think obscures our ability to study intralocus sexual conflict. We still see some strong signatures, but it's not complete. These selected animals adapted clearly to the novel genetic background and also partially to the clone generator females they were mating with. And this improved fitness, interestingly, was mediated through mating success and reduced mate harm, but not so much sperm competition, which has been implicated in the past. Lastly, we found evidence for adaptation to the cytotype and the Y chromosomes that we were using. And while this is not identical through the replicates, there's a really interesting story there. And if you're attending the in-person conference, I would highly recommend Mindy Barudi's poster. Two takeaways I want to leave you with, if we still have a second. Future work along these lines, which is becoming more and more common, needs a lot more rigorous control. And we need to be studying a much larger number of base populations. A consequence of a tight-knit research community has meant that most of the populations studied are derived from Larry Harshman's LH populations, which means that we're not even very general about Drosophila melanogaster, forget intralocus sexual conflict in general. Right, thank you so much.
Great. Thank you so much for a great talk. If anybody has questions, please send them through the Q&A or the chat, and we will move on to our next speaker to stay on time. And Yu Ying, you should be able to share your slides.
Hello.
Hi. Hi.
stage fright. Okay,
No
so
worries.
I'm going to share my screen.
Great. Looks good.
Right, then I'll get started. So hello, everyone. I'll first introduce myself. My name is Yu Ying. I was born in 1998 in Chongqing, China, and grew up in Shanghai, China. Then I went to the United States to study liberal arts at Haverford College. And then I first went to Montreal in 2017, went to another conference, and then I got interested in coding and started doing bioinformatics. My professor sent me to external projects at other universities because that was not taught in my college. And that was a good decision because later we got hit by COVID and I have to do my thesis remotely anyway. Then I graduated virtually and went to UPenn. And I didn't know what I want to do with my life at that time. So I went to actually started working at a more permanent position at Penn Dental, but I felt unfulfilled there and decided to actually do something else. I went back home to work on animation in a studio that produced this work, if anyone is familiar with it. However, I really realized that I better thrive in science. So I went back to a startup in medicine and got accepted to a EU program. I went to the Netherlands, and because of all the adventures and moving and failures in careers, and the sun that was setting at 4 p.m. every day, I got clinically depressed. So, however, in the next rotation, I went to Germany, which is more southern, and I got a very nourishing lab environment. And I was able to use the help from everyone to piece myself together again. And this talk will be about the project that I did in Germany. So constructing high resolution landscape. Resolution recombination landscape from thousands of pollen cells. What is a recombination landscape? It describes the rates of recombination along a chromosome, and it is calculated by imposing the frequency of meiotic recombinations over the genomic distance on the chromosome. And the genomic distance reflects the resolution of the recombination landscape. The recombination landscape is a carefully orchestrated product of evolution. It is, if we study the recombination landscape, we're able to discover the correlations between intrachromosome features with recombination events along a genome, as well as interchromosome features with recombination. And to study these things, we first need resolution in the recombination landscape so that we can correlate recombination events with these intrachromosome features. And we also need throughput so that we are able to generate recombination landscapes for multiple chromosomes or individuals or even populations. However, traditional methods are low in both resolution and throughput. For example, cytological methods are only able to visualize and visualize a few markers along a chromosome and pinpoint crossovers at a very low resolution. And methods such as pedigree methods can only requires the sequencing, the growing and the sequencing of tens, if not hundreds, of individual offsprings and genomes. And this not only requires a high amount of effort, but also can only sequence viable zygotes that become viable offsprings. So how do we actually generate recombination landscape in real time during the production of gametes? That is our method, which also has high resolution and high throughput. We're able to recover more than 100 markers per genome, resolving the recombination landscape to 1 megabase, which is one of the best standards currently. And we're able to generate a bioinformatic pipeline to generate recombination landscapes for more than 1,000 genomes at a time. And we're applying this workflow to plants because we already have animal recombination landscapes generated through the gamete method through thousands of sperms. But for plants, their gametes are hard to study because of cell walls and seed oil and other reasons. But we're able to solve this laboratory part in our team. So how do we work on the bioinformatic workflow? First, we have genetically divergent parents that generate heterozygous offspring. And after the production of gametes, we're able to have these heterozygous offspring pollen. We generate the reads from all three of them. And we're able to use the parental reads to separate the two genomes of the diploid offspring, assemble a genome that is phased, and then by aligning the two genomes, we're able to identify genomic variants, which we will from now on call markers. We will use these markers as a reference and map the offspring single cell reads onto this genome. This way, we are able to identify the markers and their genotypes in the single cell pollen genomes. By looking at comparing the single cell genome of the pollen cells, where with the reference haplotype, we're able to identify switches of haplotypes and therefore possible crossover locations. But we can't really say that these locations are real crossovers because reality is always more complex. We don't get a clear-cut picture, but instead we get these very complicated graphs. And we can't really assign crossovers at this high number to such a genome. So what we do is we rely on a probability model to calculate the likely identity of each segment and assign these segments identities, and then we are able to detect or call crossovers at more reasonable regions. Still, sometimes we are relying on very few markers to identify the haplotype of a segment, and we cannot really trust these results, and we have to remove these segments. And as a result, we're also not able to say if crossovers has happened in this region, and we have to remove such chromosomes. So we do have to lose data, but eventually, since we sequenced thousands of pollen, we still have a large number of pollens or barcodes for each chromosome. We calculate the frequency at different locations on the chromosome, impose the frequency of recombination events on genomic windows, and finally, we're able to generate the recombination landscape. So we used our workflow on Brassica rapa, which is a crop species and a close relative of Arabidopsis, and we're indeed able to recover our landscape at a high throughput. We are able to, so let's backtrack a little to what single cell sequencing is if people are not aware, not familiar with it. Basically, the single cells are assigned barcodes, and each cell will receive a unique barcode, and each cell will merge with this unique barcode and become an oil droplet. Each droplet is sequenced and as with their content fragmentized. So the result is short reads connected by barcodes, and we're able to use the barcode to identify the origin of these reads, therefore identify single cell reads. And usually one droplet will have one barcode with one cell, but sometimes one barcode may capture no cells or capture some background noise, or sometimes the background will, sometimes the oil droplet will capture two cells. So these things we're able to identify them and throw them out. And after filtering, we have more than 1,000 pollen cells for the production of our landscape. We're also able to recover the landscape at a high resolution. We had more than 100 markers per single genome. And since we also have these low quality barcodes, which represent likely this type of barcode that do not have a lot of markers because it does not capture a lot of chromosome material. After that, we are able to pinpoint recombination events within 1 megabase because, yes, when we are assigning segments, we are not really able to assign the segments assign the haplotype of segments to a distance that is longer than the most adjacent, shorter than the most adjacent markers. So we have to rely on the maximum marker distance between the two markers that are most farther apart to determine our recombination landscape resolution. And we used 1 megabase. However, it is still possible to increase our resolution and get a smaller window size given the type of our data. And finally, we're able to identify about 10 crossovers per barcode. So Brassica rapa has 10 chromosomes, and Brassica rapa has 10 chromosomes, and the reason we get less than 10 crossovers per genome is likely because in a previous step that we threw out some of the chromosomes. So we have this peak less than 10, but that is okay. Finally, we divide this number of crossovers by 1 megabase window that we calculated from the previous step, and we get the recombination landscape of Brassica rapa. So here we can see the recombination landscape of the 10 chromosomes. And we're able to see the classic U shape of these recombination landscapes, where recombination rates are higher towards the end of the chromosomes and lower towards the middle, which are likely centromeric regions. However, we're also able to see some weird patterns where the U shape does not have the concave in the center. That is likely because these chromosomes have centromeres that are not at the center. So from this, another Brassica rapa study, the genetic map shows that the centromeres are near one end of the chromosome. So that is actually reflected in our recombination landscapes. And for this one that is more extreme, it has an acrocentric chromosome with a centromere very close to one of the ends. And we also have a confirming recombination landscape. So in short, time allowing, I will give a summary of our methods. So we have these maternal short reads from the parents, paternal short reads from the, yes?
Sorry, that's your 12 minutes, so you have 2 minutes left.
Yes, and okay, I need 30 seconds for this. And we have this offspring long reads. Then we assemble this offspring genome with the offspring long reads using the parental reads to phase this genome into the two genomes of the diploid offspring. Then we have the single polar reads from the gametes of this offspring. We align the single polar reads to the genome. Then we get aligned reads, but we also align the two genomes together to get genomic markers to genotype the single polar genomes. And we are able to call crossovers with the haplotype switches. And finally, by calculating the frequency of crossovers onto divided by genomic regions, we're able to calculate the recombination landscape. So finally, thank this BEAM or my European study program for funding and my wonderful experience, and thank my research group and support group. From them, I learned that one person science is a community, and we need nourishing environments, and it's okay to struggle sometimes, but you're not alone, and we're all in this, and we're still able to do good work, and we can support each other. And my pet, which we call Tar because we're bioinformaticians. So thank you, everyone.
Great,
Any
thank you very much.
questions?
Thank you, Ying, for a great talk. If you have questions for Ying, please put them in the Q&A or the chat. And Megan, if you want to start sharing, get you going.
Y'all seeing the right screen?
Yes, absolutely. And we can hear you.
Okay.
Yeah, whenever you're ready.
Just trying to get my own presenter view.
Ah.
Now are you seeing the presenter view?
We're seeing your presenter view, yeah.
That's all right. Alrighty, so my name is Megan Motley. I work on sex determination in the brown anole, Anolis segrii at the University of Georgia. So today, I'm going to start my talk by talking about the absolutely striking diversity that we see throughout the animal kingdom between the species, like the different sexes of the same species, whether this is differences in feather color or fur color, appendage that's used for mate preference, sexual selection, et cetera, size, all of these morphological differences that we see between males and females is underlying a species' sex determination system. So in sex determination, we have a bipotential gonad that is undifferentiated and has a 50-50 shot essentially to develop into either a testis or an ovary. And this balance between these gametes, right? So we've got our ovary making eggs or our testis making sperm. These two gametes forming offspring are going to be absolutely fundamental to species survival and reproductive success in the animal kingdom. If you don't produce offspring, your species will not survive. So in sex determination, we often see a sex determination initiation cue received in early embryonic development. So your bipotential gonad is going to receive that cue, and that is going to initiate gonad differentiation to proceed, giving rise to either that male testis or that female ovary. We know a lot about how this works in mammals. We know a bit about how it works in birds, but we really don't know how these mechanisms work in other groups. You know, we know a bit about fish, but we really know almost nothing about reptiles, which is what I study. So Anolis lizards, they are a super diverse genus of over 400 plus species. In this image I have here, you can see kind of this striking diversity in size, color, and some of the morphological differences. Anoles live in various habitats. These are called ecomorphs in terms of where the anole lives. So our crown giant ecomorph are going to be these big, huge anoles that live, in the tops of the trees versus our trunk or trunk ground anoles that are going to live on the trunks of trees. And then we have some, and these adaptive radiation events have created this giant diversity. But one thing that all of these super diverse lizards have in common is actually genetic sex determination. So in Anolis, we have this ancient XXXY system that's been derived from a single ancestral autosomal pair. And this has arisen approximately 160 million years ago. So down here, we'll see that split off between the acrodonts and the pleurodonts under the umbrella of the Iguania group. And this acrodont-pleurodont split happened about 157 million years ago. And this is where that XY system emerged. So this XXXY system is conserved across all 400 species of Anolis and other related squamates, including most iguanas and other pleurodont lizards. So to identify a primary sex determination gene, I'm using the brown anole, Anolis segrii, as our model. The Menke lab has done a lot of work in the last few years to get the genomic and genetic resources up for these guys, so we can really use them as a really strong model system. So when we're looking for primary candidate sex determination genes, we've had to dig through the literature and kind of figure out like what are these primary sex determination genes and other groups look like when they have these ancient XXXY systems. A really good example of that is in mammals. So we knew from the start we were really looking for a gene that was located in the most ancient region of the Y chromosome, expressed early in XY gonads, right? So our males have to be expressing this early on. And then we're expecting it to be conserved across pleurodonts because all pleurodont lizards have this or I should say most pleurodont lizards have this XXXY system. And then we also needed to keep in mind that if there was an XY gene pair still around, that the Y gene would likely be highly divergent from our X gene. So in 2019, when I started my PhD, we only had a few XX reference genomes. We had the 2011 green anole genome, and then in 2022, the Menke lab published, in collaboration with the Geneva lab, a new XX genome of the brown anole, but an XX genome, as I'm sure you guys are all thinking, doesn't have a Y chromosome. So we started, the lizard here in this picture, I went out to Florida, I picked it up, and we have sequenced his genome. So we used PacBio Hi-Fi sequencing and Hi-C sequencing and scaffolded using phase genomics. We got 30x coverage, 15x coverage for our X and our Y. And what this has really revealed to us is that the Y has undergone massive gene loss, as to be expected. It is very gene poor and quite degenerate. So our X-specific region, this part down here in this light orangey color, has about 1,100 genes, whereas the Y-specific portion down here only has about 340 genes. So much less genetic content than the X. And as you'll see here marked in orange, we do know from some of our previous work in the green anole that the ancient X and the ancient Y are going to be in these sort of areas in orange. And again, that's going to come into handy when we're thinking about what genes can be sex determination genes. So we have our assembly, it looks good. What else are we going to do? Well, we need some RNA sequencing because we really need that transcriptome data to bump up and find these candidate genes. So in the anole, the gonadal ridge, which is going to be this sort of white or yellowy part in this diagram I have here, forms on top of the mesonephros, which is sort of that big pink tissue. And so what I did for my stage 7, 8, and 9, which is a time of development, those were, the gonads were actually taken off the mesonephros, pooled, and just the gonads are used in our RNA sequencing pipeline. For stage 5, the stage 5 mesonephros without gonad was used as a non-gonadal tissue, and that'll come into play when I get into some of my data. So from the assembly, we have found that there are only 5 ancient genes remaining in the most ancient region of the Y. So again, we're looking for genes that are expressed early in XY gonads, conserved across pleurodonts, and then that Y divergence is going to be significant in XY pair. So out of the 5 genes that are remaining, we've got CCDC74B, RSPH14, GNAS, UB2L3, and RPL6. So some of these genes are not conserved in iguana, they're not expressed in early embryonic gonads, and they lack sequence divergence. So they're really not, they don't look quite like a good candidate for sex determination. And so really what we were left with was this gene RPL6 on the Y. And so let's dig into kind of what that looked like. So RPL6Y is a primary sex determination gene in anolis. So RPL6 is a ribosomal protein. On paper, it doesn't look that exciting in terms of sex determination, but we know from my transcriptome data that it is being expressed in early XY gonads. The Y version, there is an X version. So RPL6Y has about 70% sequence identity to RPL6X. So it's checking all of the boxes there on divergence. And then in my preliminary results, we have found that RPL6X and Y are present in 11 other pleurodont species. These are genomes that either our lab is collaborating on or are publicly available. So a really strong candidate for sex determination. And so what does this expression look like? So RPL6Y is highly expressed in the XY gonads in early development. So what we have here is normalized read counts as a proxy for level of expression, and then stage and tissue type. These 2 tissues here in the front in this light blue are going to be non-gonadal tissues. So we've got that stage 5 mesonephros, we've got a stage 7 forelimb from some of our other work, and then we've got stage 7 gonad, stage 8 gonad, and stage 9 gonad. And what you can see is that in stage 5 mesonephros or forelimb, we have very low expression of RPL6Y in male samples. But at stage 7, right, we're getting a very high increase of expression of RPL6Y that seems to sort of start to taper off a bit as we go on through development. So again, that's going to be that check mark of like, it is being expressed early in XY gonads. So then we wanted to see, okay, well, what is RPL6X doing then? And we actually found that there's sort of this very interesting phenomenon happening where in stage 5 mesonephros and forelimbs, again, our non-gonadal tissues, in our Y sample, in our XY samples and our XX samples, it's much more highly expressed. And then when we look at our gonad specific samples, you'll see that in the females in these XX samples and in the XY samples, we're seeing a way lower expression of RPL6X. So now we're thinking, okay, well, if RPL6Y is being highly expressed when males are developing, we're also seeing that this RPL6X seems to be being decreased in a gonad specific way. So doing some digging on the actual structure of our gene, we noticed that there is RPL6 is up here in blue. We noticed that there is overlap with this gene PTPN11 with their exons. So that is interesting. We didn't know much more. We started to dig into this a bit. And so what we see is that we looked at our splicing patterns. What we see between PTPN11 and RPL6 in non-gonadal tissues looks totally normal, right? We've got our RPL6 splicing patterns and our PTPN splicing patterns. But then when we looked at our gonad samples, so this is going to be an XX female gonad versus an XY male gonad. And what we're actually seeing is a pretty striking mRNA splicing event happening. So what does this mean? Well, we looked a bit more, and there's actually a gonad specific promoter for PTPN11 that generates an antisense RPL6X exon. So gonad specifically, this PTPN gonad specific promoter splices into RPL6. And we've decided to call this antisense transcript for RPL6X and anolis Astra. So what is Astra doing while RPL6X is being decreased and RPL6Y in males is being increased? Well, what we see again with our males and our females, so this is stage 5 mesonephros, male and female, non-gonadal tissue, non-gonadal tissue, we have almost zero expression of Astra. When we get into our stage 7, 8, and 9 gonads, again, we're seeing that very high, we're seeing high expression of Astra in male and female gonads specifically. So while RPL6X is being decreased in XX and XY gonads, Astra seems to be being increased, and that's allowing for that RPL6Y to be increased. So as a big picture, this looks like, so in dark blue, we've got Astra. So again, very little expression in our non-gonadal tissues. And then we're getting that increase at stage 7 in that gonad specific manner. We're seeing RPL6Y again, lower in the non-gonadal mesonephros, non-gonadal forelimb, and then we're seeing that expression elevated in the stage 7 gonads. And then what we can see with RPL6X in this light blue, again, is that RPL6X in XY gonads is going to be highly expressed in mesonephros, in forelimb, but is being decreased in the gonad. So what does this mean? It's a ribosomal protein, and there is mounting evidence that there is sex specificity in ribosomal proteins. So these two papers are kind of starting to hit the edge. So a male germ cell specific ribosome controls male fertility, and then ribosome heterogeneity in Drosophila melanogastric gonads through paralog switching. So both of these papers do a really nice job of sort of starting to chip away at that, this idea that ribosomal proteins seem to be specific and sex specific. And so as my project progresses, we will be trying to tease out that mechanism with the ribosomal protein, RPL6. So future directions, I'm going to continue analyzing available pleurodont genomes to understand if that mechanism is conserved. We know from some other work that it does appear to be Astra and RPL6X and Y seems to be conserved in the green anole. And then we'll be utilizing CRISPR gene editing to functionally validate RPL6Y as the initiator of primary sex determination in asagri. So I have started to do this, and we are now in the screening period trying to hopefully get some sex reversed males. And then I will be using mRNA and C2 to determine the locality of expression for RPL6 and Astra. And with that, I want to say thank you guys so much. Thank you to the organizers. Thank you guys for having me. Special thanks to the Monkey Lab, in particular, my mentor, Doug, Sungdae Park, Britta Osman. Thank you guys so much.
Great. Thanks so much, Megan. That was a lovely talk. So that brings us to the end of our session. And because we're one speaker short, why don't we can open up questions